Today I am going to explain about nomenclature of enzymes and classification of enzymes. So first one coming to nomenclature of enzymes. Nomenclature. So nomenclature means provide a particular name to specific enzymes. So this called as a nomenclature of enzymes. So these nomenclature will useful to identify the enzyme and function properties of enzymes. So first one. So provided names to enzymes by three methods. These are trivial nomenclature. Trivial or old nomenclature. Then second one, recommended nomenclature. Then third one, systematic nomenclature. So provided names to enzymes by three methods. So first one, trivial, old nomenclature, recommended nomenclature, then systematic nomenclature. So first one coming to trivial old nomenclature. According to this method, provide names to enzymes by source of material. By source of material to enzyme, then family, then function, then action. So according to this method, by this method provide the names to enzymes by source of, then family, function and action. So first one, source. So for example, papain. So the enzyme name papain according to source of the material. So carica papaya plant. Source of this enzyme is a plant papaya, carica papaya plant. That's why provided name to this particular enzyme. That means which enzyme work on a specific substrate. So called as a papain. Next one, bromelain. So bromelain enzyme, the source of material is a pineapple. Pineapple. So pineapple belongs to Bromeliaceae family. That's why this enzyme called as a bromelain. Next one, based on the function, number of dehydrogenase enzymes. Dehydrogenase enzymes, glyceraldehyde 3-phosphate dehydrogenase, etc. Next one, nucleases, lipases, peptidases, proteases, amylase, etc. etc. So based on the function and action, provide the names to enzymes. This method is known as a trivial or old nomenclature. So in trivial old nomenclature, provide names to enzymes based on source, family, function and action. So next one, recommended nomenclature. In recommended nomenclature, provided names to enzymes by following the substrate, nature of chemical reaction, then some suffix words. So according to this nomenclature, each name of the enzyme contain substrate name, then nature of a chemical reaction. Nature of chemical reaction, nothing but the name of a chemical reaction. Next one, suffix word is. Suffix word is. So generally the suffix word "ase" used for enzymes to root word of the substrate. But some enzymes names have not the suffix word "ase", like a pepsin, then trypsin, then chymotrypsin, etc. But these are valid names. So according to recommended nomenclature, each enzyme name contain the word of substrate name. Next one, type of chemical reaction, then a suffix word is. Example, succinic dehydrogenase enzyme. Succinic dehydrogenase enzyme. In this enzyme, substrate is a succinate. Substrate is a succinate, nothing but succinic acid. Next one, nature of chemical reaction. Type of chemical reaction is a dehydrogenation. Then suffix word is. So succinic dehydrogenase enzyme. Generally, succinic dehydrogenase enzyme located in inner membrane of mitochondria play important role in Krebs cycle. Krebs cycle through oxidation reaction. By the activation of this enzyme, catalytic nature of this enzyme, succinic acid converts into fumaric acid. So remove the hydrogens from succinic acid. These hydrogens are transferred to reduce the coenzyme FAD plus. So FAD plus converted into FADH2. So according to recommended nomenclature, each enzyme name contains substrate name. So for example, in succinic dehydrogenase succinate, succinic acid. Next one, class type of the chemical reaction. Nature of chemical reaction: dehydrogenation, removal of hydrogens from succinate or succinic acid, transfers to coenzyme FAD. Then suffix word is. Next, third one, systematic nomenclature. It is a modern type of nomenclature. According to these nomenclature, each enzyme name contain the word of substrate name. Next one, cofactor type, nothing but coenzyme, nothing but coenzyme name. Next one, IUB, class of chemical reaction. IUB, class of chemical reaction. Then suffix word is. So systematic nomenclature is a modern type of nomenclature. According to this nomenclature, each enzyme name as a substrate name, the word of substrate name. Next one, coenzyme name, cofactor, non-protein part. Next one, IUB, class of a chemical reaction, type of chemical reaction. Then suffix word is. Example, succinic FAD plus dehydrogenase plus the suffix word is. So total enzyme is a succinic FAD dehydrogenase enzyme. Succinic FAD dehydrogenase enzyme. Here substrate name is a succinate, succinic acid. Then coenzyme is a FAD. Then type of the chemical reaction is a dehydrogenation, removal of hydrogens. Then suffix word is. So systematic nomenclature is a modern type of nomenclature. This nomenclature method recommended provided by IUB, International Union of Biochemistry. According to this nomenclature, each enzyme name as a substrate name. Next one, coenzyme, cofactor type name or Next one, chemical reaction, nature or class of chemical reaction. Then a suffix word is a ase. Next one, classification of enzymes. Classification of enzymes. So classification of enzymes based on action. So based on action, classify the enzymes two types. These are exo enzymes. Next one, endo enzymes. So generally enzymes are organic substances, that means organic chemicals, proteinaceous substances. So all enzymes are proteins, but all proteins are not enzymes. These are biological origin, maybe synthesis enzymes inside the cell, but this work outside the cell or inside the cell. So based on these action, enzymes are two types, exo enzymes, extracellular enzymes, then endo enzymes, intracellular enzymes, extracellular and intracellular enzymes. So these extracellular enzymes synthesize formation inside the cell, but this work outside the cell. This move out from inside the cell to outside, perform outside. Why? Because maybe this source of raw material for chemical reaction may be existing outside the cell. That's why this move from inside to outside, from the source of origin to source of work. This type of enzymes is known as exo enzymes, extracellular enzymes, digestive juice. So digestive juice contains these are extracellular enzymes. So lysozyme enzyme, amylase enzyme, all digestive enzymes belongs to these exo enzymes, extracellular enzymes. Next one, endo enzymes. These endo enzymes synthesizing inside the cell, then work also inside the cell. So number of metabolic enzymes, respiratory enzymes, that means mitochondrial enzymes, then chloroplast enzyme, photosynthetic enzymes, etc. Mitochondrial metabolic enzymes. Next one, based on the structure. Based on the structure, enzymes are two types. So these are simple enzymes. Second one, complex enzymes, compound enzyme or polar enzymes. So based on the structure, enzymes are two types. So these are simple enzymes. Second one, complex, compound or polar enzymes. So first one coming to simple enzymes. In simple enzymes, only proteinaceous part is present. Non-protein part is absent. Only protein part is present. Nothing but amino acids. Amino acids. So these amino acids are building block of proteins. Proteins is nothing but polymer of amino acids. So this protein part is a amino acid. By the hydrolysis of these simple enzymes, only gives the amino acids. Why? Because only these are proteinaceous enzymes. Only protein part is present. Non-protein part is absent. This type of enzymes is known as a simple enzymes, pepsin, trypsin, chymotrypsin, etc. Examples. So pepsin, then trypsin, chymotrypsin, etc. So these are the simple enzymes. Only protein part is present. Non-protein part cofactors are absent. Next one, complex compound enzymes are holo enzymes. In these enzymes, protein part along with protein part non-protein part also present. So protein part, then non-protein part, non-protein part. So protein part is called as a apoenzyme. Protein part is called an apoenzyme. Non-protein part cofactor. Non-protein part cofactor. So complex compound enzymes contain protein part and non-protein part. So protein part called as a apoenzyme. Non-protein part called as a cofactor, including these known as a holoenzyme. Holoenzyme is a nothing but protein part and non-protein part cofactors. Holoenzyme. So apoenzyme is a protein part, cofactor is a non-protein part. Next one, coming to cofactors. So cofactors are non-protein part in a holoenzyme or complex compound enzymes. These cofactors are three types. First one, prosthetic group. Second one, coenzyme. Third one, metal ions. Cofactors are three types. So first one, prosthetic group, then coenzyme, metal ions. So prosthetic group and coenzyme both are organic. Both are organic. It is a inorganic. Prosthetic group and coenzyme both are organic nature, then metal ions are inorganic, that means salts, mineral ions. Then prosthetic group is a organic nature, organic compound. These are tightly bound with the apoenzyme, that means a protein part while chemical reaction. After completion of chemical reaction, this prosthetic group remains with the apoenzyme, so called prosthetic group. Example, heme. Heme group in peroxidase and catalase enzyme. Heme group in iron. Heme group in peroxidase and catalase, iron metal and Fe. So prosthetic group. So prosthetic group is a organic compound. These are tightly bound with the apoenzyme, protein part. After completion of catalytic reaction also these are remains with the protein part. Example, heme in peroxidase and catalase enzyme. Next one, coenzyme. Coenzyme also organic in nature, but this loosely bind with apoenzyme. Maybe after completion of chemical reaction, catalytic reaction, these may be separated from protein part. Apoenzyme is called coenzymes. So coenzymes are organic in nature. These are loosely bound with apoenzyme, protein part. So after completion of this catalytic reaction, these may separated from this protein part, apoenzyme. So coenzymes are generally vitamins. Coenzymes are generally vitamins. These are derivatives of B vitamin. These are derivatives of B vitamin. Mostly all coenzymes are derivatives of B vitamin. Examples are TPP. So TPP, thymine pyrophosphate, vitamin B1. Vitamin B1. So function generally decarboxylation. Decarboxylation. So TPP, thymine pyrophosphate, vitamin B1. So generally function is a decarboxylation. Next one, FAD and FMN. So FAD and flavin adenine dinucleotide flavin mononucleotides. So these are coenzymes. These are coenzymes in our electron transport system, mainly in electron transport system, aerobic respiration. So FAD is a prosthetic group or coenzyme in complex 2. Then FMN is a prosthetic group or coenzyme in complex 1. So these are very useful to transfer the electrons, oxidation as well as a reduction. So vitamin B2, riboflavin. Function is oxidation as well as a reduction. Function is a oxidation as well as a reduction. Next one, NAD. So NAD also coenzyme, nicotinamide adenine dinucleotide, nicotinic acid, niacin, nicotinic acid, niacin. Function also oxidation as well as a decarboxylation. Next one, coenzyme. Coenzyme A. Coenzyme A, pantothenic acid. Pantothenic acid. So coenzyme A, pantothenic acid. Biological acetylation. Function is biological acetylation. Function is biological acetylation. Next one, coenzyme B12. Coenzyme B12. So B12 vitamin function is isomeric reactions. Isomeric reaction. So coenzymes all are vitamins. These are mostly derivatives of B vitamin. Examples are DPP, thiamine pyrophosphate, vitamin B1. Function is decarboxylation. Then FAD, FMN, vitamin B2, riboflavin, oxidation as well as a reduction. Then NAD, niacin, nicotinic acid, oxidation as well as a reduction. Then coenzyme A, pantothenic acid, the function is biological acetylation. Then coenzyme B12, which means B12 function is isomerism, isomeric reactions. Next one, metal ions. So metal ions generally inorganic in nature. So metal ions. So metal ions generally inorganic nature. Metal ions are essential to protein part apoenzyme for catalytic nature. Without these metal ions, some enzymes cannot catalyze some reactions. So metal ions Na plus sucrase enzyme. So in sucrase enzyme, Na plus sodium act as a cofactor. Next one, K plus pyruvic kinase enzyme. Next one, Fe plus cytochrome oxidase. Next one, peroxidase. Catalase. So Fe plus cytochrome oxidase, peroxidase and catalase. Next one, Ca plus succinic dehydrogenase enzyme. Succinic dehydrogenase enzyme. So Krebs cycle. Next one, Mg2 plus. So Mg2 plus act as a cofactor in all polymerase reactions, all kinase reactions, all isomerization or phosphorylation reactions. Example, UBP carboxylase and oxygenase. UBP carboxylase and oxygenase. Then PEP carboxylase, RUBP carboxylase, C3 cycle, then PEP carboxylase, C4 cycle. All phosphorylation reactions, polymerization reactions, etc. Next one, Cu2 plus cytochrome, cytochrome C oxidase. So cytochrome oxidase, cytochrome C oxidase, Cu2 plus. Next one, Mn2 plus IAA oxidase, MO nitrate reductase, then Zn2 plus carbonic anhydrase, then carboxy peptidase, metal ions. So these metal ions act as a cofactors. Best example, Na plus sucrase engine, K plus pyruvic kinase engine, Fe plus cytochrome oxidase, peroxidase, catalase, Ca plus succinica, dehydrogenase, Mg2 plus all polymerases, phosphorylase enzymes, RUBP carboxylase in C3 cycle, PEP carboxylase in C4 cycle, Cu2 plus cytochrome C oxidase, Mn2 plus IAA oxidase, oxime synthesis, MO nitrate reductase, nitrogen metabolism. Next one, Zn2 plus carbonic anhydrase, carboxy peptidase. Next one, classification of enzymes. Classification of enzymes according to IUB, International Union of Biochemistry, now known as IUB-EMN, International Union of Biochemistry and Molecular Biology. International Union of Biochemistry, now known as International Union of Biochemistry and Molecular Biology. So IUB classify the enzymes into 6 classes. IUB classify the enzymes into 6 classes. Then each class classified into 4 to 13 subclasses. 4 to 13 subclasses. So these 6 classes are: first one, oxido reductases, dehydrogenases. So first type is a oxidoreductases, dehydrogenases. These type of enzymes catalyze the reaction oxidation as well as a reduction. Nothing but transfers the electrons by using the oxygen or without using oxygen. In presence of oxygen, remove the hydrogens. These hydrogens are transfers to coenzymes. So results of these are, these coenzymes are reduced coenzymes like NAD2, NADH, FAD2, FADH2. This type of reactions is known as a oxidoreductases enzymes. So these enzymes catalyze both the oxidation as well as a reduction. Examples: peroxidases. So these type of enzymes catalyze the both oxidation as well as a reduction. Examples are dehydrogenases, oxidases, reductases, oxygenases, catalases, peroxidases. So transfers the electrons from one substrate to another substrates. Is reduced is apostrophe oxy Nice. So after completion of chemical reaction, this substrate S is a oxidized, then S appears to be reduced. This is due to transfer of electrons from one substrate to another substrate. So transfer of electrons from S apostrophe is to S. So S1 is a oxidized, S apostrophe is a reduced. So by use of oxygen or without use of oxygen, in presence of oxygen only, alcohol plus NAD, alcohol dehydrogenase enzyme, aldehyde NADH2. So here transfers the electrons from alcohol to NAD plus. So NAD plus reduced to NADH2. So it is oxidized to aldehyde. This entire reaction catalyzed by alcohol dehydrogenase enzyme. So first class of enzymes are oxidoreductases, dehydrogenases. These type of enzymes catalyze the reaction oxidation as well as a reduction. Transfers the electrons from one substrate to another substrate. Examples are dehydrogenases, removal of hydrogens, oxidases, reductases, oxygenases, catalase and peroxidases. So these are the examples. So transfers the electrons from one substrate to another substrate. So due to this one, it is reduced. So here transfers the electrons from alcohol to NAD plus. Transfers the protons, hydrogens from alcohol to NAD plus. NAD plus reduced to NADH2. This reaction catalyzed by alcohol dehydrogenase enzyme. Next, second one, transferases. Transferases. So these type of enzymes catalyze the reaction transfer of groups one substrate to another substrate. This is called transferases. So these type of enzymes catalyze the chemical reaction transfer of groups from one substrate to another substrate. So this is called transferases. So second type of enzyme is a transferases. These type of enzymes catalyze the reaction transfer of the groups from one substrate to another substrate. Examples are all kinases. Next one, phosphorylase, phosphotransferases. So here transfers the group from one substrate to another substrate. So here X group transfer from S to S apostrophe. Like it is a reduced condition. So glucose plus ATP from the glucose 6-phosphate, ATP converted into ADP. This reaction catalyzed by hexokinase enzyme. Hexokinase enzyme, glucokinase enzyme, hexokinase or glucokinase enzyme. In glycolysis, first reaction is a phosphorylation. That means transfers the inorganic phosphate from ATP to glucose. This reaction catalyzed by hexokinase enzyme, glucokinase enzyme. So glucose phosphorylated to glucose 6-phosphate transfers the one inorganic phosphate from ADP, adenosine triphosphate. So ATP converted into ADP molecule. Next, third class of enzyme, hydrolysis. Hydrolysis. So third class of enzyme is a hydrolysis. This type of enzyme catalyze the hydrolysis. That means break the large molecule converted into simple molecule with the help of hydrogen and hydroxyl groups. So these type of enzymes catalyze the break the bonds of ester bonds, then ether bonds, then peptide bonds, then glycosidic bonds, C minus C bonds, C minus halide bonds, then P minus N bonds. So hydrolysis, these type of enzymes catalyze the reaction hydrolysis. That means break the larger molecule, convert into simple molecule, break the bonds, existing bonds in polymer of compounds with the help of hydrogen and hydroxyl groups. These enzymes break the bonds of ester bonds, ether bonds, peptide bonds, glycosidic bonds, then carbon-carbon bonds, C halide bonds and P-N bonds. S first group H2O. So after completion of a reaction SH, then S first group OH. Sucrose. Sucrose is a disaccharide, so non-reducing sugar. These sucrose are converted into glucose and fructose. These reactions catalyzed by sucrase or invertase. Sucrase or invertase. This type of class of enzymes is known as a hydrolysis. Sucrose is a disaccharide, non-reducing sugar. These contain two types of monosaccharides, glucose and fructose. This glucose and fructose are joined by glycosidic bonds. So these type of enzymes, sucrase or invertase enzyme, break the glycosidic bond. Then sucrose convert into glucose and fructose. Then glucose and fructose then participate into glycolysis. That means respiration. First step of a respirative process. So hydrolysis. These hydrolyse enzyme catalyze the reaction hydrolysis. So break the large molecules, then convert into simple molecules. The N type of the bonds will break ester bonds, ether bonds, peptide bonds, glycosidic bonds, CC bonds, C halide bonds, then P-N bonds. Lyases. Then fourth class of enzyme is lyases. These type of enzyme catalyze the reactions formation of double bonds. Formation of double bonds by removing groups. So these type of enzymes catalyze the reaction formation of a double bonds by removing the group or removing of double bonds. Removing of double bonds by addition of groups. Addition of group. So these type of enzymes catalyze the reaction formation of double bonds by the removal of groups or removal of groups by the addition of groups without hydrolysis, without the help of the water molecules. Here may be help of the water molecule, here without help of the water molecule, without hydrolysis, without oxidation, without the reduction, without consume or use the ATP molecules. So lyases enzyme catalyze the reaction formation of double bonds by removal of groups or removal of double bonds by addition of groups without hydrolysis, oxidative enzymes, reductive enzymes, without consume the ATP energy. So here formation of a double bonds. Fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate converted into DHAP, dihydroxyacetone phosphate, then glyceroldehyde 3-phosphate. Generally, fructose 1,6 bisphosphate is a 3-carbon compound. Key to sugar and glycolysis fifth step. This reaction catalyzed by aldolase enzyme, then converted into 3-carbon sugars. Thiosa phosphate DHAP, dihydroxyacetone phosphate ketose sugar, then G3P glyceroldehyde 3-phosphate aldose sugars. Examples of these enzymes are aldolases, then hydrases, then carboxylases, then decarboxylases, then enolases. So examples for lysases, aldolase enzymes, hydrases, decarboxylase, carboxylase and enolase. So fourth class of enzymes are lyases. So these type of enzymes catalyze the catalytic reaction, formation of double bonds by removal of groups or removal of double bonds by the addition of groups without hydrolysis, oxidation, reduction, without using ATP molecules. Examples are aldolases, hydrases, carboxylase, decarboxylase and enolases, removal of water molecules. Example, fructose 1,6 bisphosphate is a 6-carbon ketose sugar. By the catalytic activity of aldolase converts into 3-carbon compounds DHAP ketose sugar, then G3P aldose sugars. Next, fifth class of enzyme, isomerases. So these isomerase enzymes catalyze the reaction isomerase of one compound into isomeric compound. Intramolecular shift of groups or inorganic phosphate one carbon to another carbon. This type of enzymes is known as isomerases. These type of enzymes catalyze the chemical reaction isomerism. That means one compound converts into another isomer compounds. That means change the groups from one carbon position to another carbon position. Before and after isomerization, both the compounds has the same amount of energy, same number of bonds, same number of carbon compounds, but the arrangement of the groups are different condition. Before isomerization, the groups what are changed after the isomerization may be in different position. So due to this isomerization, these groups may be arranged in different carbon position. But before isomerization, after the isomerization, this both compounds has the same amount of energy, same number of carbons, same number of bonds. Examples are epimerases, then chartamerases, then methymerases. So examples are epimerases, chartamerases and metamerase enzymes. X, then S apostrophe Y. So after is X, then S apostrophe Y. So glucose 6-phosphate. Glucose 6-phosphate. So convert into fructose 6-phosphate by the activity of isomerase enzyme. Example for isomerase, glucose 6-phosphate, 6-carbon aldose sugar by the isomerase enzyme convert into fructose 6-phosphate ketose sugar. That means aldose sugar convert into ketose sugar, but the same number of carbon, 6 carbons are present, but arrangement may be different condition in aldose sugar and ketose sugar. This both are have the same amount of energy, same number of bonds. So before the isomerization, after the isomerization. Sixth one, ligases. So these type of enzymes catalyze the reaction, linking of the molecules, joining of the molecules, or joining of the bonds by consuming ATP energy. This is known as a ligases. So these ligases enzyme catalyze the reaction, linking of the molecules, joining of the molecules, joining of bonds, formation of bonds by consuming ATP energy is called ligases. These enzymes also known as a synthetases. Succinate thiokinase enzyme. Examples are all synthetases and succinate thiokinase enzyme. Yes, sir. So S minus S first protein AGP. Glutamate plus ammonia by consuming AGP here from the glutamate, then release the water molecule. So glutamate is a amino acid, then add the ammonia group by consuming ATP molecules here from the glutamate. Amides release the water molecule. So generally amides are modified amino acids, contains the large amount of ammonia compared to amino acids. So ligase enzymes, these enzymes form the bonds of C minus O bonds, C minus S bonds, C minus N bonds, then PO bonds. So these ligases enzyme form the bonds of C minus O bonds, C minus S bonds, C minus N bonds, and PO bonds. These enzymes also known as a synthetases. Example, succinate thiokinase enzyme. Next one, each enzyme name contains the 4 diseases. Each enzyme name contains the 4 digits. That means 4 codes are present. Each enzyme name contains the 4 code according to IUB, International Union of Biochemistry. Each enzyme name contains the 4 codes. So first code class, then second code subclass, then third code subclass, then fourth code serial number, individual enzyme number. So according to IUB, International Union of Biochemistry, each enzyme name contains 4 codes, like a 4 disease. So first code denotes the class of the enzyme, then second code subclass, third code subclass, then fourth code individual number, serial number of enzymes. So this class denotes the nature of chemical reaction. Nature of chemical reaction. It is a type of a substrate. Then type of a coenzyme or nature of a coenzyme. It is a ordinal enzyme member. Ordinal. So first code denotes the class, nature of chemical reaction. Second code subclass, nature of a substrate. Then third code subclass, nature of a coenzyme or cofactor, non-protein part. Next one, fourth code individual enzyme number, ordinal serial number of enzymes in subclass. Example, glucose. 6-phospho transferase. Glucose 6-phosphotransferase. So enzyme code is 2712. Example, glucose 6-phosphotransferase. For this enzyme code is 2712. So 2 represents the class, 7 represents the subclass, 1 represents the subclass, then 2 serial number of the enzyme. So nature of chemical reaction, nature of substrate, then nature of non-protein part, then serial number in subclass. So each digit in enzyme code separated by dots. Separated by the dots. So in code each digit separated by dots. So classification of enzymes.